So, welcome back to the virtual classroom. Here we are in my glorious kitchen. Today, I'm going to talk very briefly about the process of protein synthesis, which is effectively just the steps of transcription and translation that take us from DNA instructions to an actual physical protein that expresses genetic material, right? And so like the protein is what actually gives our characteristics their characteristics. And so this whole process is the foundation of our understanding of genetics and how gene expression works and how heredity works. So without understanding these steps, we can't really understand genetics. So this is where we're going to start. So the whole process starts with our DNA molecule. And our DNA is simply a long chain of nucleotides in a very specific order. And the order in which the nucleotides occur are what controls the specific outcome of protein and ultimately our characteristic itself. And so within a species genome, such as a human, right, there are small variations in that code order, but overall, there's a very generalized pattern in which these things show up. Right? And so we all look pretty much the same, right, when you compare us to like a chair or a sofa or a cabinet or a cat or a hammer or a hot dog. Right? Us humans, we all look the same. Now, if you look in detail, there's a lot of variation in our individual traits, and those variations come from those small, slight changes in nucleotide order. Okay? And so the process of transcription is the first step of protein synthesis. Right? And we are opening up the DNA molecule. Right? We take the double helix, we split it open, and we are reading the very specific code of any individual gene. Now, in the DNA, there are long stretches of junk DNA. It's just complete trash. There's no coding in there at all. And then specific sections are genes. Right? And so the gene for eye color, for instance, just as an example, will always be on the same place, on the same chromosome for all of us. Right? And so pretend it's on chromosome 7, spot 4332. It will always be in spot 4332 for each of us humans. If we have blue eyes, it might be AGGGCCCCG. If we have brown eyes, it might be AGGGCCCCT. Right? Slight differences give us our variations. And so transcription starts with an enzyme called RNA polymerase. Okay? RNA polymerase opens up the DNA, right? Breaks the little hydrogen bonds between the nucleotides, splits it open, okay? And it reads it, right? So the RNA polymerase finds an A in the DNA. It reaches out, it grabs a U from the surrounding area and sticks it on, right? They're complementary. It shifts down a level. Okay? It finds a G, reaches out, grabs a C, goes down, finds a T, reaches out, grabs an A, plugs it on. And it builds this RNA strand that is a complementary strand to this DNA. Okay. So RNA polymerase moves down the DNA strand from the 3' to the 5' direction. So our RNA, our mRNA, is built from 5' to 3'. Okay. And so the direction is important because of the way that they latch on. Right? And so the shape of the RNA polymerase will only latch on to the 3' end, right? Because the shapes line up properly. Okay? And when it adds that complementary piece, it changes that shape ever so slightly and pushes it down. Right? And it moves and moves and moves, okay, until it hits a particular region of this gene that is shaped in a way that makes it pop off. RNA polymerase floats away. My RNA is now made, that floats away, and then the DNA, those hydrogen bonds can close back up. Right? They're like little magnets. Close right back up. DNA is back to normal, happy. I got my cute little RNA floating around that represents this gene. Okay? And transcription is done. So transcription is over, and I got this RNA floating around in my nucleus because it just popped right off my DNA, so it's fresh. It's a baby RNA. Right? This RNA is what we call pre-mRNA. It's about to be a big adult mRNA, but it's not there yet. It needs a little bit of modification. Right? And we'll go into the modification steps in the gene regulation section of this unit. Okay? But for now, just know that we have to modify this bad boy a little bit before it becomes our actual mRNA. Okay? So once those modifications are done, that mRNA floats out of the nucleus and out into the cytoplasm, right? And it very quickly reaches the endoplasmic reticulum, where it will engage with some of those ribosomes. Right? So the mRNA is going out there to meet some ribosomes. The ribosomes, if you recall from our cell organelle unit, start as two different pieces, a large subunit and a small subunit. Right? When that mRNA floats around and clicks into the small subunit, it changes the shape a little bit so that now the big subunit, the large subunit, can fit on. And they become like a hamburger bun. Right? So they get this closed hamburger bun around that mRNA. Now the mRNA, if you recall, is complementary to the original DNA strand, meaning it has a very precise order of RNA nucleotides. Okay, and that precise order is very important for the process of translation that's about to happen. Okay. Within the ribosome, there are three little compartments. Okay. The mRNA clicks into the first compartment, right? And the ribosome processes the mRNA in groups of 3 nucleotides. Okay. So there's these 3 nucleotides sticking out of the first compartment. Okay. This is what we call a codon. Okay. Codons are only existing in name, right? There's no physical delineation between one codon and the next, right? It's just that the ribosome has enough room for 3 nucleotides. 3. Okay, so that's my codon. All right. In the top of the chamber, there's a slot here. So I got my 3 nucleotides sticking up like this. Okay, I got my chamber above it. Now I need a tRNA with an amino acid attached to it. Okay, so there's a bunch of different tRNAs floating around with slightly different shapes. Okay, and each tRNA is associated with a specific amino acid. Okay, and so in our bodies we utilize 20 amino acids. Okay, and every single time I have a very specific shaped tRNA, it's always going to go with the same amino acid. So let's pretend it's leucine, because leucine sounds cool. I like leucine. Leucine. Okay. So I got my leucine attached to my tRNA. Okay. It's attached to the top of the tRNA. Down at the bottom of the tRNA, there are 3 nucleotides sticking out. These 3 nucleotides are going to be complementary to a very specific codon set. Right? And in this case, it's going to match these. So pretend my codon was AUG, right? My anti-codon, which is the tRNA part, is going to be UAC, okay? And they're going to be complementary. So they're going to line up nicely, okay? So this TNA happened to get in here, boom, lined up and clicked, okay? That clicking changes the shape of the ribosome a little bit. Right? And it bumps the large subunit forward, okay, boop, okay. And then my amino acid pops off my tRNA, okay, which changes shape again, pops my subunit back to in line, right? That moves everything forward. My tRNA is now naked, no more amino acid attached. It's ejected, flies back out into the cytosol of the cell, okay? And my amino acid is now stuck on the little ejector port on top of the ribosome. Okay. As this happened, right, the RNA has now moved down a little bit, the mRNA, and so now I have a new codon set, looking for a new tRNA with a different amino acid, and the same thing will happen. And it slides down and down and down and down the mRNA, lining up different codons with different anticodons for the tRNA, which are associated with amino acids. And at the top of the ribosome, we're building these long polypeptide chains of amino acids. Okay? They're the precursor to what is going to be our final protein. translation. So now I've reached the end of my mRNA. My ribosome slide all the way down, reaches the end. It hits what's called a stop codon. The ribosome says, all right, I'm done. It disengages its two subunits and they float away. Okay. Meanwhile, I have this polypeptide chain, right? And as it's been coming out and getting longer, it's been folding up into itself, right? And making a very unique shape. Okay. And now, depending on the order of the amino acids, which is determined by the order of the mRNA nucleotides, which was determined by the order of the DNA nucleotides, right? This protein is going to fold in different ways, very specific ways. Right? And that folding is going to be what gives this protein its function. We will go over protein folding in depth. It will probably have its own video. Protein folding is cute. I like protein folding. It's not hard. Makes sense. It's logical. There are like a few levels of it, but as its own topic, it's no big deal. So I got my folded up protein. It's finalized, right? It's no longer a polypeptide. It is now a protein because it has its final shape, and now it is ready to rock, right? And so depending on its shape, right, it has different functions. So let's go back to the eye color example. If this is shaped properly, right, let's say it has this little thing sticking out right here. Okay. When light hits it, boop, it's going to catch certain frequencies of light and release other frequencies. Okay. And in this case, in the human eye, when our genes work properly, we have brown eyes. Right? We have brown pigments in our eyes to protect our eyes from the sun. So this properly shaped protein catches those harmful sunlights and doesn't let our eyes get hurt by the sun. Okay, these would be brown eye proteins. With a slight modification in the DNA sequence, right? I have a modification in my RNA sequence, and I'll have a modification in my amino acid sequence in my protein. So my protein will be folded a little bit differently. Let's pretend that it doesn't have that part jutting out anymore. Okay, so this shape of protein can no longer catch the appropriate wavelengths of light. Okay? And this is how we get blue eyes, right? Our brown proteins are broken, and so we don't catch the sunlight coming in. Okay? And so ultimately that small change in the DNA sequence resulted in a change in the shape of my protein, resulted in a change in the characteristic that is represented in the entirety of my beautiful existence. Okay? So that's it. Protein synthesis done. Yeah, cool. Bye.